The second part of the Forensic Biology Screening Workshop deals with blood. What is blood? Blood is a slightly alkaline fluid made up of water, cells, enzymes, proteins, glucose, hormones, organic and inorganic substances. Blood circulates throughout the body. It supplies nutrients and oxygen to the body and removes waste. Blood cells are made in the bone marrow through a process called hemochromatosis. It starts as a stem cell known as a blast, and some immature blood cells stay in the bone marrow to mature. Others travel to other parts of the body to mature, example lymph nodes, spleen, and liver. These cells mature and differentiate into several classes of cells: red blood cells, white blood cells, and platelets. Red blood cells, also known as erythrocytes, have no nucleus and therefore not useful for DNA analysis. They are approximately 6 to 8 micrometers in size. They are approximately 45% of the total volume of blood, and they are the most abundant cell in the blood. Red blood cells are disc-shaped cells which make up 99% of the cells in the blood. They are the principal carriers of the red-colored hemoglobin molecules. Hemoglobin is an iron-containing protein and binds about 97% of all oxygen in the body. This is an example of what red blood cells look like under a microscope. Hemoglobin contains 4 subunits of globular proteins with heme. Heme groups contain iron, which binds oxygen. Arterial blood is bright red when hemoglobin carries oxygen. Venous blood is dark red. Veins appear blue in color due to optical effects of light when hemoglobin lacks oxygen. Here is an example of what a hemoglobin molecule looks like. And this is an example of the chemical structure of heme containing iron. Notice the iron Fe is in the center of the heme molecule. White blood cells, also known as leukocytes, are produced in the bone marrow. White blood cells have a nucleus which is useful for DNA analysis. They are a vital source of defense against external organisms. White blood cells also clean up dead cells and tissue debris that would otherwise accumulate and lead to problems. There are 5 classes of leukocytes: neutrophil, eosinophil, basophil, monocyte, and lymphocyte. Here is an example of red blood cells and white blood cells next to each other. Notice that the white blood cells are larger and they contain a darkened region inside. This is the nucleus which contains the DNA. Platelets are irregularly shaped, colorless bodies produced in the bone marrow. Their sticky surface lets them, along with other substances, form clots to stop bleeding. They are only active when damage occurs to the circulatory system walls. Here is an example of hemostasis. Platelets in the bloodstream combine with collagen fibers to form a platelet plug to repair a damaged blood vessel. Plasma is the liquid portion of blood. It is composed of water, proteins, and electrolytes. Blood cells and platelets are suspended in plasma. Plasma regulates osmotic pressure, and it is the transport medium for glucose, lipids, hormones, oxygen, but not as much as heme, clotting factors, and waste. Serum is a clear liquid that is left after blood coagulates. Serum is plasma without the clotting factors. Now let's go over the forensic significance of blood. First, hemoglobin, which is found in red blood cells. It has a peroxidase-like activity that can cleave hydrogen peroxide. The blood group antigen, which is also found in red blood cells, are bound to the red blood cell membrane and they dictate the ABO groups. DNA is found in white blood cells, and these are found in cells with a nucleus. And proteins, which are found in the plasma, are basically serum used in species testing. Here are two common terms that will be used a lot in forensic testing. First, presumptive tests. These indicate a substance is present and they are not specific. The second term is confirmatory tests. These confirm a substance is present and is very specific. Now let's go over some common presumptive tests that are used when testing for blood. We have the Kastle-Meyer test, also known as phenolphthalein, the Leuco Malachite green test, also known as LMG, Hemostasis, and Luminol. Some other tests that are used are Tetramethyl Benzene, TMB, benzidine, orthotoluidine, and orthotoluidine. Most presumptive tests are catalytic tests, and they are based on the fact that hemoglobin and some of its derivatives exhibit a peroxidase activity. Tests based on this property are generally named after the compound undergoing oxidation, like benzidine or TMB, or the discoverer, Kastle-Meyer. The oxidant, hydrogen peroxide, oxidizes a colorless reagent to a colored reagent. Heme catalyzes this oxidation by cleaving an oxygen from hydrogen peroxide. This chemical equation is showing the reaction that is occurring during the Kastle-Meyer reaction. You have hydrogen peroxide and your colorless reagent. When they are mixed together in the presence of a heme catalyst, will give a pink reagent. This reagent is now pink because of the removal of the hydrogen groups from the hydroxide on the colorless reagent. Now let's go over how to make the Kastle-Meyer reagent. First, you have phenolphthalein plus potassium hydroxide and zinc. You reflux, boil, and condense. During this process, you're reducing the phenolphthalein to phenolphthalein. Oxygen is removed and combined with OH from potassium hydroxide, and this boils off as water, and this becomes a colorless solution. The working solution is phenolphthalein, ethanol, and zinc pellets. The phenolphthalein reagent is stored in an amber bottle. This is because light can affect stability. There are zinc pellets at the bottom of the bottle, and these bind free oxygen and prevent oxidation. The working solution can be kept at room temperature, and the stock solution should be refrigerated. Now let's go over how to perform the Kastle-Meyer test. Place a small cutting, swabbing, or extract of the suspected blood stain on filter paper, or swab the stain using a slightly moistened swab. The Kastle-Meyer test is a three-step test. Step 1. Add 2 to 3 drops of ethanol to the stain or swabbing. This will increase the sensitivity by cleaning the area around the hemoglobin, better exposing the heme. Step 2. Add 2 drops of reagent and then wait for approximately 5 seconds. This step aids in ruling out false positives due to the presence of chemical oxidants such as rust, because if they were present, a reaction would occur at this point. Step 3. Add 2 to 3 drops of 3% hydrogen peroxide. If an immediate color change to pink occurs, then the test is positive for the possible presence of blood. If no color change occurs, then either blood is not present or it is in too limited quantity for the test to detect. Note that the swab will eventually turn pink even if negative over time due to the nature of oxidation reactions. Limitations of the Kastle-Meyer test. Sensitivity. Stains diluted to 1 in 10 to the power 5 to 1 in 10 to the power 6 and allowed to dry gave a positive result. Specificity limitations. The test can weed out false positives between steps 2 and 3. However, some chemical oxidants and vegetable peroxidases gave false positive results. The test also will not detect differences in animal or human blood. And it is relatively stable if the reagents are stored separately and refrigerated. Here is a video of the Kastle-Meyer test showing how the test is performed.
The Kastle-Meyer test is used as a preliminary screening test for blood. After putting on appropriate protective gear, the analyst takes a small sample of the suspected blood stain, placing it on either a swab or filter paper. A few drops of ethanol and phenolphthalein are now added to the stain. After waiting to ensure that no color develops at this stage, 2 or 3 drops of hydrogen peroxide are added. After a few seconds, an intense pink color will begin to develop as seen here. If no pink color is seen, the test is negative for hemoglobin, which would be indicative of blood.
Leuco-Malachite green is another reagent that is used for presumptive testing of blood. Here is an example of the Leuco-Malachite green reaction. Notice that you're starting with Leuco-Malachite green, which is colorless. Notice the center carbon has a hydrogen on it. Plus hemoglobin and 3% hydrogen peroxide, you get an oxidized form of the malachite green, which turns a blue-green color. Notice now that the center carbon no longer has the hydrogen on it. Making Leuco-Malachite green. First you have malachite green plus acetic acid plus water plus zinc. You reflux, boil, and allow to condense, and the solution becomes colorless. The working solution is the LMG reagent plus zinc pellets. Leuco-Malachite green is stored in an amber bottle with zinc pellets, and it is relatively stable at room temperature. How to perform the Leuco-Malachite green test. Place a small cutting, swabbing, or extract of the suspected blood stain on filter paper, or swab the stain using a slightly moistened swab. Step 1. Add 1 to 2 drops of the LMG reagent. Note if there is a color change. If there is a color change, the test is considered inconclusive. Step 2. Add 1 to 2 drops of hydrogen peroxide. Note the results. If the color change goes to a deep green or blue, the test is positive for the possible presence of blood. If there is no color change, the test is negative. Limitations of the Leuco-Malachite green test. It was found to be sensitive only up to 1 to 1000. As for specificity, chemical oxidants and vegetable peroxidase did give false positives, and it will not detect the differences in animal or human blood. Its stability was similar to that of Kastle-Meyer. This video shows how to perform the Leuco-Malachite green test.
Leuco-Malachite green, also known as LMG, is a presumptive test for blood. After putting on appropriate protective gear, the analyst swabs the suspected blood stain with a swab or clean filter paper. Next, 1 to 2 drops of the LMG reagent is added. The analyst should note any blue-green color change at this step. If a blue-green color change occurs, the test is considered inconclusive. If no color change occurs, a couple drops of hydrogen peroxide are added. The analyst again notes any blue-green color change. This time, a color change indicates a positive result, and no color change indicates a negative result.
Another presumptive test used is called Hemostasis. These are reagent strips. In one bottle, there are about 50 reagent strips, and they are stored at room temperature. The test is based on the peroxidase activity of hemoglobin. The reagent on Hemostasis is diisopropyl benzene dihydroperoxide and 3,3,5,5 Tetramethyl Benzene, TMB. The color change ranges from orange to green, possibly blue with higher concentrations of blood. How to perform the Hemostasis test. Step 1. Slightly moisten the pad on the tip of the strip with water. Step 2. Rub the damp pad on the stain in question. Note a color change within 60 seconds and compare to the chart on the bottle. More green indicates more hemoglobin. Limitations of Hemostasis. It is sensitive up to 0.015 to 0.062 milligrams per deciliter of free hemoglobin. Its specificity is affected by chemical oxidants and vegetable peroxidases, which will both give false positive results. It also will not be able to detect the differences in animal or human blood. and it is stable for approximately 1 year. The expiration date is stamped on the bottle. Here is a video that is showing how to perform the Hemostasis test.
Hemostasis, originally designed for testing blood and urine, are used as a presumptive test for the presence of blood. After putting on appropriate protective gear, the analyst rubs the dampened pad onto the stain in question. Within 60 seconds, a color change should take place. A weak reaction is indicated by orange, and a strong reaction is indicated by blue. A final color ranging from orange to blue indicates a positive result. No color change indicates a negative result.
Now let's go over luminol, another reagent that is used to test for blood. You have hydrogen peroxide plus luminol in the presence of a base and a catalyst gives 3-amino phthalate. The 3-amino phthalate gives off light. How luminol works. The ion in hemoglobin acts as a catalyst to cause a reaction between the luminol and hydrogen peroxide. The luminol loses nitrogen and hydrogen and gains oxygen. This results in 3-amino phthalate, which is energized and emits light. Reagents needed to make luminol are luminol, which is 3-amino phthalate hydrazide, sodium perborate, distilled water, and sodium carbonate. A spray bottle works best for testing with luminol because luminol is usually used to test large areas. Luminol must be made each time that is used. To perform the luminol test, spray the luminol directly onto the stain in question. This test needs to be done in the dark to see the luminescence reaction, which can last for approximately 15 seconds. If the stain emits a light, then the test result is positive for the possible presence of blood. If there is no reaction, the result is negative. Limitations of luminol. It is sensitive up to dilutions of 10 to the power 6 to 10 to the power 8. It is the most sensitive presumptive test. It does, however, have many false positives: bleach, metals, chemical oxidants, and vegetable peroxidases. It will not detect the differences in animal or human blood. It is also very unstable. There is only an 8-hour limit. Luminol is mostly used at crime scene because it can dilute out a stain, possibly too much for DNA analysis. It is used more for blood spatter and crime scene reconstruction. Tetramethyl Benzene, or TMB, is another reagent that is used for the detection of blood. In its reduced form, it is colorless. When it is added to hemoglobin and 3% hydrogen peroxide, it turns to a green color. This is the oxidized form. In the oxidation process, hydrogen molecules are removed from the nitrogen at either end of the tetramethyl benzene molecule. Tetramethyl benzene is relatively easy to make. 3,3,5,5 tetramethyl benzene is added to glacial acetic acid. This is a lot easier to make when compared to KM and LMG reagents. TMB is stored in an amber bottle because light affects its stability, and it must be refrigerated between uses. It is only good for about 1 week. To perform the test, place a small cutting, swabbing, or extract of the suspected blood stain on filter paper, or swab the stain using a slightly moistened swab. First, add 1 drop of TMB solution, then add 1 drop of 3% hydrogen peroxide, and look for a color change. If the stain turns bluish-green, the test result is positive for the possible presence of blood. If there is no color change, then the test is negative. Limitation of TMB. It is only sensitive up to dilution of 1 to 10,000. And it is not as specific as the Kastle-Meyer test. You get false positives to vegetable peroxidases, bleach, and potassium permanganate. Also, it will not detect the differences in animal or human blood. The reagent is very unstable. It can only be used for 1 week maximum, and it loses sensitivity by a factor of 10 after 1 day. TMB is also known to be a mutagen. Some other tests that are used for the detection of blood are benzidine, orthotolidine, O-tolidine, which is 3,3-dimethyl benzidine. It has an increased sensitivity but a decreased specificity, and it has the same stability when compared to the Kastle-Meyer test. However, it is rarely used due to safety concerns because it is carcinogenic. Now let's go over species testing. First, some definitions. An antigen is a soluble protein that causes the production of an antibody. It is usually foreign to the body. An antibody is a Y-shaped immunoglobulin produced by B lymphocytes in the bone marrow, and it is produced in response to an antigen. This is a diagram of an antibody. Notice at the ends there is an antigen binding fragment. This fragment has a specific shape, so the antibody will only bind to antigens that are complementary to that shape. Antigens and antibodies bind together to create a lattice network of molecules. This lattice network then becomes insoluble and precipitates out of solution. To make antibodies, a host animal like goat or rabbit is injected with human serum. The goat or the rabbit will make antibodies in response to this serum. Then, the heart blood is collected and tested for these antibodies. The sample extracts are made from stained areas of interest, and from nearby unstained areas. These are called substrate controls. Note that the use of unstained controls is a fundamental principle in forensic immunological testing. The original precipitin reaction was carried out by layering a solution of antibody on top of a solution of stain extract in a tube. This mixture was left for a period of time to allow the development of a precipitin band at the interface. This is referred to as the tube method and is still used in a few laboratories today. Octo is another precipitation method. The method was first published in 1948 and uses radial diffusion of the antigen and antibody through an agar gel. To perform it, extract the stain in a microcentrifuge tube with saline. The extract should be straw-colored. Stain and control samples are loaded in the outer wells, and a drop of anti-human antiserum is loaded into the center well. The process is repeated for antisera to other species, such as dog, cat, and cow. This may include the species from which the antiserum was obtained, example, rabbit. The plates are left at room temperature or 37 degrees Celsius for a suitable period, which can range from a few hours to overnight, and the serum proteins and antibody molecules diffuse outward from the wells. A precipitin band is formed when the diffusing stain contains proteins that are recognized by IgG molecules in the diffusing antiserum. The precipitin band is sometimes clearly visible to the naked eye, but it is normal to stain the plates with amido black, Kamasi blue, or other general protein stains to enhance sensitivity and clarity. If a white line occurs between the samples, then the test sample is positive for human antigen activity, or whatever species is being tested. If no white line occurs, then the test sample is negative for human antigen activity, or whatever species is being tested. The Octo test is only sensitive to a dilution of 10 to the negative 1. It gives false positives with detergents and tannic acid, and it can give false negatives if there's an excess of antibodies or antigens, because then the lattice will not form and no precipitate will be seen. These diagrams are showing what is occurring in the Octo double diffusion. In the first plate, you have the antibody in the center well and the different antigens in the surrounding wells. There is also a well that will usually have a control. Then, the antibodies and antigen diffuse outwards from the wells, and they can give different types of results. If they intersect, that is called non-identity. if you have a spur being formed, that indicates partial identity, and if you have an arc, that shows a full identity. Here is an example of a positive result of an Octo-Loney plate. The line of identity is where the precipitin forms between two wells. Here is a short video on how to perform the Octo-Loney test.
The Octo-Loney test allows the immunological identification of a protein to assist in identifying human blood. After putting on appropriate protective gear, the analyst begins by labeling the plate. The analyst may choose to make their own plate or purchase a pre-configured plate, as is done here. On one side, the middle well is filled with anti-human serum, and on the other side, the middle well is filled with normal serum. Next, each corresponding well is filled with the appropriate sample. A known human standard, distilled water as a negative control, and the sample in question. The plate is then left overnight at room temperature. The next day, the analyst observes the plate for precipitin bands. A positive result is shown by precipitin bands forming a continuous arc of convergence between the antiserum well and the sample well.
Crossover electrophoresis is a variant of the Octo-Loney test, which uses an electric field rather than diffusion to move the extract and antibody through the gel. Crossover electrophoresis for species identification is conducted using agar at a pH of 8.6. Stain extracts are loaded into wells arranged in a line at the cathode end of the plate, and the antiserum is loaded into wells at the anode end. During electrophoresis, the electric field drives the serum proteins towards the anode, but the IgG molecules, which are essentially neutral at this pH, are driven to the cathode by the process of electroendoosmosis. The antigen-antibody precipitation occurs at the interface between the two rows of wells. Electroendoosmosis occurs because the supporting medium acquires a negative charge. If free, the negatively charged molecules would migrate to the anode, but this is not possible because the agar is immobilized on the plate. Instead, the effect is countered by positively charged water molecules migrating to the cathode. The migrating water molecules carry any dissolved neutral molecules such as IgG with them. The ABA card hemotrace tests for human hemoglobin. If human hemoglobin is present, it reacts with a mobile monoclonal anti-human hemoglobin antibody. This forms a mobile antigen-antibody complex, and then this migrates to the T-zone. In the T-zone, there are polyclonal anti-human hemoglobin antibodies. This forms an antibody-antigen-antibody complex. The antibodies were tagged with a pink dye, and upon aggregation at the T-zone, a pink line is seen. There is also a control zone, which has immobile anti-immunoglobulin, which binds excess anti-human hemoglobin antibodies, also to form a pink line. To perform the ABA card hemotrace test, first extract a cutting from the stain in approximately 300 microliters of buffer. You leave for 1 to 5 minutes. Then, add 150 microliters of the sample to the S well of the test card. Wait 10 minutes, then read results. A pink line in the T and C zones represents a positive. A pink line in only the C zone will represent a negative. Some important limitations of the ABA card hemotrace. The high-dose hook effect. This can give a false negative. This occurs with excess hemoglobin, which binds to the stationary anti-human hemoglobin antibodies in the T area. This prevents the mobile antibody-antigen complex from binding, and therefore results in no pink line, which will be read as a negative. This is an example of a false negative. Also, the ABA card hemotrace test is not a confirmatory test for human blood because it gives positive results with ferret blood. Advantages of the ABA card hemotrace test is that it is highly specific, reproducible, sensitive, and quick and easy to use. Similar tests exist for other types of serological samples, such as a test for semen or the familiar pregnancy test. Here is a short video on how to use the ABA card Hemotrace test.
The ABA card Hemotrace is a testing device used to aid in the possible identification of human blood. After putting on appropriate protective gear, the analyst takes a swab or small cutting and places it into a sterile microcentrifuge tube. Extraction buffer is then placed into the tube and vortexed. The tube is then set aside to incubate for 1 to 5 minutes at room temperature. The ABA card is then removed from the sealed pouch and labeled properly. 4 drops of the sample are then added to the sample well, labeled S, on the ABA card. After 10 minutes, the results are read. The presence of 2 pink lines, one in the T area and one in the C area, indicate a positive result. The presence of only 1 pink line in the C area indicates a negative result. If no pink line is present in the C area, the test is invalid.
The Serotek HemeDirect hemoglobin assay also tests for human hemoglobin. It is similar to the ABA card Hemotrace test. A negative is seen by a single line in the C zone. A positive is 2 lines, one in the C zone and one in the T zone, and an invalid result will be no lines in either of the zones or a line only in the T zone. Another test for blood is looking for the formation of hematin crystals. Teichmann published this method in 1853. Blood is treated with a solution of potassium bromide, potassium chloride, and potassium iodide in glacial acetic acid, and is heated to react with hemoglobin. The first reaction converts the hemoglobin to hemin, and then the halides react with the hemin to form characteristic brownish-yellow rhomboid crystals. The test went through many modifications. One drawback was that the test did not always work, even when the presence of blood was known. Another test for blood is the formation of hemochromogen crystals. There are many variations to this test. Takayama described his method in 1912, and it became the preferred method. His name is commonly used to describe the reagents and procedure he devised, and also the hemochromogen crystals obtained. Blood is treated with a saturated dextrose solution, sodium hydroxide, pyridine, and water. Ferrous iron from hemoglobin reacts with pyridine to produce red feathery crystals of pyridine ferroprotoporphyrin. Here we have a picture showing what Takayama crystals would look like. Even though crystal tests are unique to blood, they are not human-specific, and they are not widely used anymore. What is more commonly used today are a combination of the Kastle-Meyer test and the ABA card test, and this is mainly due to time and money constraints. Also, with DNA testing and qPCR, these are more specific for higher primate blood. Controls are a very important part of any testing. There are positive controls, negative controls, and substrate controls. Positive controls are used to determine if the tests are working properly. They are a quality control check, and this is usually documented in the case file. An example of a positive control will be known blood on cloth or a swab. A negative control is used to determine if the reagents are contaminant-free and working properly. This is also a quality control check and is also documented in the case file. An example of a negative control is unstained cloth or a swab. A substrate control is used to determine if the substrate is interfering with the test. This is usually used for troubleshooting, and this is documented in the case file. An example would be an unstained area adjacent to the questioned stain being tested.